Hi, I'm Curran, and today I'm going to be talking about plant rhizosphere interactions along with Jack and John. I'm going to be defining the rhizosphere as well as talking about its structure and ecology. So the rhizosphere comes from the roots rhizo and sphere, so the root area. In three regions, it's the endorhizosphere, the apoplastic space between cells in the epidermis and cortex, as well as the rhizoplane, the area of direct contact with the root surface, as well as the ectorhizosphere, so the sphere of influence going into bulk soil a few centimeters around the plant. It's defined by diverse bacterial, archaeal, fungal, and protist communities, as you can see in these electron microscopy images up here, how densely packed the rhizosphere is with microbes, which can have a ton of positive, negative, and neutral implications for plant health related to stress tolerance, nutrient access, phytohormone synthesis, or the disease state. How dense is it? Turns out there's about 10 billion to 1 trillion bacteria per gram soil in the rhizosphere. So there's a lot of opportunity for different interactions to happen. What determines the microbial composition of the rhizosphere? Spoiler alert, it's very complicated. First, there's the abiotic factors, which determine what bacteria or other microbes are even present in the soil in the first place before we take into account the plant. So that's oxygen levels, texture, water holding capacity, cation exchange capacity, salinity, existing nutrients, and temperature. The host also plays a really big role in recruiting microbes. So vertical transmission of disease or beneficials from seeds, as well as exudates from the roots, which can attract bacteria and other microbes through chemotaxis or even activation of microbial genes for colonization. These are diverse, often low molecular weight compounds. So amino acids, sugars, fatty acids, growth factors, vitamins, organic acids, which can be species specific or more general, or dependent on development or environmental factors. Plants have even been known to release carbon compounds into the soil under nutrient stress just to try to attract more microbes into the rhizosphere in hope of getting more beneficial interactions. One example of this is the recruitment of Burkholderia species by ZMAs in Lupinus albus. So roots have been known to release oxalate associated with higher colonization rates of Burkholderia species, but it's found that only known beneficial Burkholderia species were able to metabolize oxalate as opposed to pathogens. When you knock out the gene for metabolizing oxalate in beneficials, they're no longer able to effectively colonize plants well. So that's evidence of coevolution between microbes and hosts. There's also microbe-microbe interactions, which also very much structure the microbial community. There's cooperative ones like reciprocal nutrient transfers, where bacteria depend on each other to provide nutrients they can't synthesize themselves, as well as biofilm formation and molecular signaling, which we'll talk more about later, and competitive interactions, so direct competition and production of antimicrobials. So here's some examples. You can even see it on petri dishes here grown up, bacteria on the left and bacteria against fungi on the right. You can see that they're actively producing microbes to inhibit each other. In isolation, these interactions can seem really simple. Say we have one beneficial here, it's suppressing a pathogen, and there's another neutral microbe that is upregulating that beneficial microbe. But if we get a new beneficial that maybe downregulates the neutral one, suddenly our first beneficial, there's less of it, and the pathogen is able to thrive. So would we really still consider that neutral one always a neutral microbe, or would we always consider this beneficial on the right always beneficial? It turns out when you multiply all these interactions by 1,000, 10,000 times, etc., it can be really complicated, and most microbes start to occupy a gray area between beneficial and harmful. You can almost think of the rhizosphere as a network of co-occurrence of different microbes. So you can see there's like hub taxa, which affect lots of other taxa. There's lots of connections between them, and they structure the microbiome, as well as core taxa commonly associated with the host that can be thought of likely to be co-evolved with a plant. So we're going to focus on some specific, well-characterized interactions here, but keep in mind how complex the rhizosphere really is.
Legumes in rhizobia. Why might plants need nitrogen? You might have selected one or two options. However, nitrogen is important for all these processes in the plant. As just mentioned, plants need to incorporate nitrogen in order to perform crucial growth and developmental processes. In order to obtain nitrogen, nitrogen fixation and nitrogen assimilation must occur. Nitrogen fixation is the process of converting atmospheric nitrogen, hydrogen, and ATP into ammonia. As a result, plants need atmospheric nitrogen to be converted into a usable form like ammonia or nitrate to assimilate it for other processes. Nitrogen assimilation is the process of converting inorganic nitrogen compounds such as nitrate or NH4 ammonium into other usable organic forms. Knowing that plants can't directly use atmospheric nitrogen and cannot perform nitrogen fixation by themselves, how do they obtain usable nitrogen? In some plants, like legumes, they are unable to perform nitrogen fixation and must rely on rhizobia to do it for them. Rhizobia is a group of soil bacteria that enters the cells of a host plant and helps to fix nitrogen. The relationship between rhizobia and host legumes is symbiotic, as the plants receive ammonia from the rhizobia and the rhizobia receive carbohydrates. For this relationship to occur, plants must form root nodules. Root nodules are special organs that host rhizobia and provide an anaerobic or low oxygen environment for nitrogenase activity. Here are the steps for nodule formation in plants. When legumes experience low nitrogen conditions, they want to form a symbiotic relationship. In order to do this, they release flavonoids into the soil, which are basically chemical signals to attract rhizobia. When flavonoids bind to nodulation genes in rhizobia, they then produce nod factors. Nod factors are signaling molecules that initiate a signaling cascade to initiate changes in root development. More specifically, nod factors deform the root structure into curling, which allow rhizobia to 1. Locate and proliferate in a confined area, and 2. Help degrade the surrounding plant cell walls to form an infection thread. An infection thread acts as a pathway for symbiotic bacteria to travel through within the plant. Once an infection thread is formed, rhizobia can then travel from the root epidermal cells to the root cortex cells. In the apoplast of the root cortex, rhizobia fuse into them and release nod factors again to trigger cortical cells to divide rapidly, eventually forming a root nodule for nitrogen fixation. Symbiotic mycorrhizal fungi. Mycorrhizae are a fungus that forms a symbiosis with the roots of a plant. Unlike rhizobia, which are plant-specific, mycorrhizae are non-selective as symbionts, meaning that they can form symbiotic relationships with many plants. As a result, mycorrhizal relationships are found in upwards of 80 to 90% of land plants. As a symbiont, both plants and mycorrhizae benefit from each other. Mycorrhizae receive carbohydrates, while plants, on the other hand, receive water, nitrogen, phosphorus, and other micronutrients from mycorrhizae. Hyphae come in different sizes as well. Smaller or fine hyphae provide surface area for nutrient absorption, while on the other hand, larger or coarse hyphae support and provide nutrient transport. Mycorrhizae are split into two different categories based on how they interact with the root interface. Ectomycorrhizae form a sheath around the root, and as a result, the root is not penetrated. So how do they interact with the root? They form a hardtech net. Essentially, this is a network of hyphae that colonizes the apoplast of the epidermal and cortex cells. Endomycorrhizae, on the other hand, penetrate the root cell epidermis and cortical cells. In this case, root cells are penetrated, so how do they interact with the root? Endomycorrhizae can form hyphal coils, which are coils around root cells, or they can form branch arbuscules, which are tree-like structures made of hyphae which penetrate into the epidermal and cortex cells and branch inside the cytoplasm of the cortex cells. Arbuscular mycorrhizal fungi are found in the roots of 80% of land plants. But why? It's believed that these fungi helped plants colonize the land and helped the evolution of vascular plants. Based on the previous slide, what type of mycorrhiza is this? This is an arbuscular mycorrhizal fungi because they penetrate the root cells. Mycorrhizal association steps. The steps for mycorrhizal associations with plants are extremely similar to how rhizobia interact with the host legumes. First, hyphae grow into soil pores and reach nutrients inaccessible to the plant. Then, initiation and germination of the hyphae occur. The exact mechanism isn't known, but mycorrhizae can sense CO2 or plant root exudates, which is similar to how rhizobia sense flavonoids from the plant. In ectomycorrhizae specifically, mic factors are made by mycorrhizae. These are similar to how nod factors function as well in rhizobia. Mic factors help the hyphae initiate root branching. The root is infected by the mycorrhizae in two different ways. In ectomycorrhizae, hyphae grow between dermal cells, but in endomycorrhizae, they form a hyphopodium, which is the short branch of hyphae that attaches to the external epidermis. Hyphae can then extend into the cortex and form arbuscules to reach nutrients, and as a result, a symbiotic relationship occurs. How mycorrhizae help plants. The soil around plant roots where nutrients are lacking is called the nutrient depletion zone. With the help of mycorrhizae, they essentially extend the surface area of the roots, which can reach into the soil, increasing the availability for water and nutrient absorption, which is up to a 10 times increase. Mycorrhizae can also help to stabilize the soil and can form a common mycorrhizal network where hyphae connect multiple plants. This allows for nutrient sharing, communication between distant plants, and pathogen, disease, and metal protection.
Hello everyone, my name is Jack, and today I'll be discussing the importance of rhizosphere microbes to kind of finish out our presentation and get you an idea of how these interactions are really benefiting the plant in the long term. So starting with nutrient acquisition and cycling, the main point here is that rhizosphere microbes are essential because they break down complex organic compounds to release minerals that the plants need. So even in soils that are very low in nutrients, these microbes make sure that the plants are getting what they require for healthy growth. This continuous nutrient uptake directly boosts plant health and yields and makes it a key factor in sustainable agriculture, which we'll talk about later. Plant growth and promotion is next. So this kind of is the rhizosphere microbes producing growth hormones and stress-related hormones. So these are familiar to us as we learned in class about auxins and cytokinins, and these are actually things that these microbes are producing for enhanced root branching, extension, and better access to water and nutrients for the plant. Down here, we're talking about stress-related hormones, which really are just producing productive compounds, and beneficial microbes are helping the plant better manage environmental stresses such as salinity or temperature extremes and changes. Moving on to disease suppression and biocontrol, the overall idea here is producing antimicrobial substances that protect the plant roots from harmful pathogens. With this natural defense in place, plants can focus their energy on growing and reproducing instead of fighting infections, which is just going to create a stronger, more resilient plant. That's what I'm getting into here with the optimized energy allocation, deciding where the plant wants to put its energy because it doesn't have to worry about these pathogens. As you can see, this front line of defense here is kind of giving an inhibiting effect on these pathogens, allowing for more growth. Next, soil structure and ecosystem resilience. So here we're just talking about microbial secretions and how they're playing a significant role in improving the overall soil structure. They bind soil particles together, enhance water retention, reduce erosion, and just create a better environment for plant roots. This improved soil structure supports nutrient uptake and long-term fertility. Essentially, you can think of it as an insurance policy for the plant. It helps it adapt to various stresses and changing conditions without any detrimental effects coming from those changing conditions. Finally, future perspectives, I just want to kind of look ahead and talk about emerging omics technologies like genomics, which are revolutionizing our understanding of plant microbe interactions. So researchers are even starting to explore microbiome engineering, which involves selecting or designing beneficial microbial communities. And this could lead to increased plant health, reduced chemical dependencies on things like pesticides, and more sustainable farming practices, which gets us into my last point, which is just a question for you guys. How could these emerging omics technologies really change how we view farming? And we've learned a lot in this presentation how these rhizosphere interactions are really helping the plant against these adverse conditions. But how could omics technologies take this even further? Something for you guys to think about.